Hello and welcome to Simplified. My name is Gurjot
And my name is Michael.
and this is episode 3.
In this episode, we spoke to Collin Ewald. Collin is a professor of health sciences here at the ETH Zurich, and he does research on getting old. Or not.
Or not, yeah. He studies how aging works. And I learned a pretty interesting thing from him was that it's not just genes or DNA structure that go on aging,
Yeah.
it's also neurons.
Also neurons.
Yeah, like it's the way you think that makes you older. So it's at the end of the day, you are as old as you think you are.
Actually, Collin covered a huge range of topics.
Yeah, he covered a huge range. He pretty much covered everything. Like, if you stick to the science, he covered the history of aging research. He told us how present-day research works, where they're trying to go with it. And then he also talked about the financial burden of aging research and also the philosophical or social concern. Would you want to live to 1,000 years? And in the end, he even gave us a lab tour.
He showed us the worms they use to study aging. It was really cool, and you can even see this on our website. Ladies and gentlemen, this is Aging 101.
Okay, could you tell us a little bit about your educational background and what got you into doing the research that you do today?
Well, I was originally born in Switzerland, and did my undergraduate at the University of Basel, and then for my master's I went to the Friedrich Miesche Institute, it's also in Basel, and there I was working with Nancy Heinz and Joy Alcedo. And Joy Alcedo, she was interested how the environment affects aging, and she was working with a little organism called Synoptitis elegans. It's a little roundworm. It's 1 millimeter in size. It's great for genetic studies. And there, what they have found, so this worm has like 302 neurons, and you can apply certain neurons with a laser. And if you apply the right neuron, you can increase the lifespan of the worm, which really fascinated me, because if you think about it, it's like somehow neurons govern like the rate of aging, right? And at the same time, I read some article where they looked at people and they trained actually people to meditate for like 20 minutes for 6 weeks, and then they measured their gene expression, right, with microarrays. And what they found is that meditating for like 6 weeks changed their gene expression, right? So in other words, you could control with what you're thinking your gene expression, and then combined with the finding in C. elegans that you, with certain neurons, you can regulate the rate of aging. I got really fascinating with the aging process and neuroscience.
What do you mean by gene expression?
So you could look when genes are expressed, right? So you have it on the DNA, and then genes are expressed and they're transcribed into an mRNA, and that goes into a protein, right? So you can measure the levels of mRNA, so the relative levels. And so basically what they have done, the people meditated and they took blood samples. And so the gene expression in their blood samples changed, and which was for me amazing.
Looking at the mRNA, that's an indicator of aging.
Well, the gene expression level constantly changes, right? So with different things, but you could use the expression levels, of course, also look at aging. So there's certain patterns that change with aging. So people are doing this also, yes.
That fascinated you, and that's when you decided to move into this research.
Exactly. So I was fascinated by neurons and aging. So I did my PhD in neuroscience in New York, City University in New York, and then to go even further into aging, I went to the Charles M. Diabetes Center, Harvard Medical School, to do my postdoctoral training, and then became an instructor in medicine there, and now I'm here to continue my research.
Awesome.
Okay, we'll start off with a very simple question that's pretty much on everyone's mind. What is the secret to long life?
Well, I mean, I think nobody really knows, right? And I mean, this really, this, as you pointed out, this is a question that people were asking for centuries, right? I mean, people were looking for the fountain of youth, like Pantaleon and or like the alchemists trying to have the philosopher's stone, and then also Chinese emperors, they took things like mercury and things like that to think to make them live longer, and they died. And so this was all before really science hit off, right? So when science hit off, we had the scientific method, and then people starting to think, well, can we apply like rigorous science to the aging process, right? And that's how it really started. And the breakthrough there came again from a model organism. So as I told you, this nematode C. elegans lives around, say, 3 weeks, right? And so Cynthia Kenyon found a mutation in a single gene that was able to double the lifespan of this organism, which was really crazy, because if you think about aging, it's such a complex process, right? And then a mutation in a single gene, and then the whole organism lives double as long, right? And so that really hit the whole thing off. So then we had somehow something to make an impact in real research, gather data, right? And then people have started to look whether, okay, this is true for a little worm, right? Is this also true, say, in mice, right? In mammals? And then they looked at the same gene in mice, and when they did genetic manipulations, right, it also was sufficient to increase lifespan. And so there the increase is usually around 15 to 30%, which is also very good. Now, the cool thing really started when people were starting to look at centenarians. Centenarians is like 1 out of 6,000 people is all over the age of 100 years, right?
Yes.
Yeah.
So, and those centenarians, actually, people that pass 100, most of them are pretty healthy for a long time, right? They're healthy and fit, and they can do basically whatever they want. They drink alcohol, they eat lots of meat, they don't do any exercise, they smoke and things like that. They're somehow protected, right?
So they have basically a similar behavior to normal people, except they live a lot longer.
Exactly. And so when they look now at what's different on the genetic level, right? So look at their DNA, and then you compare those long-lived centenarians to normal-lived humans, right? They have variants in certain regions in their genome, right? And then they pinpointed down those variants, and one or a couple of those variants are exactly in the same genes that were found in C. elegans or in the mouse. So this is the insulin IGF-1 receptor signaling pathway. And so, things that really started, I mean, in C. elegans, that was 1993, right? And then the mouse was like 10 years later, and then as soon as we had these genomic data, right, then they really hit off. So there seems to be some relevance. What could be found in other smaller organisms has also relevance for human aging. And now, for example, Craig Venter has founded a company, Human Longevity, where they sequence just millions of genomes of people, right? And out of those different variants, they're trying to figure out which are actually maybe protective against diseases, right? Or give you a longer life. And basically, this is now the new kind of way to unlock the secret to a long life.
So basically finding the right genes that create the right effect.
Exactly. You find to pinpoint those associations, right, of these gene variants to, say, protection against cancer or Alzheimer's disease or other things, right? Well, that all means that it's an association, and as you know, correlation is not causation, right?
Yes.
And so the thing becomes now how you go about to test those variations, right? And now in biology, the cool thing is that most things are very well conserved. So important genes and pathways are very well conserved. As I mentioned, the example with C. elegans, the same pathway was conserved in mammals and in us humans, right?
Like it's similar across different organisms.
Exactly. So because you have to remember, we come from the same ancestor cell, right?
Yes.
Right, yeah.
Okay. And so the genetic code is the same, and things that are really important, they stay kind of the same. It's not
Exactly
one to
the
one,
same,
but
yeah.
it's it's very well. So if it's very well conserved, so now you could imagine you have now all those gene variants in those very long-lived people, right? And if those genes are conserved, right, you could test those variants in smaller organisms. So you could introduce the same mutation in the small organism and see how long it lives. And this is like your litmus test you have for chemistry, right? It's a functional readout for whatever association you have. You could get some evidence that it affects the aging process. Now, you have to understand that the aging process is actually very conserved and it's universal. So even if you think about single-cell organisms like yeast or bacteria, right, they also age. So for example, if you take a yeast cell, right, that buds off all the time and you keep the mother cell, with time that mother cell ages because it retains all those damages that are made to proteins or DNA, it retains in the mother cell and then gives off a young cell, right? And you could do the same thing is true for bacteria. So it's really like something that's really universal and really ancient, right? And so now scientists are trying to unlock those secrets.
So you talked about how you looked at centenarians, people who live above the age of 100. Do you also, apart from looking at their gene structure, do you also look at the reasons why this came about? Because from what I know, there's a Greek island and which has lots of old people who just party all the time and still live to above 90 and even 100 and beyond. And then there's the island of Okinawa in Japan, where there's an entire village which has the world's most number of centenarians in just one short place. And I've been to Okinawa, I worked there for a little while, and what I noticed was they have a traditional Okinawan diet, which is just tiny portions of lots of different kinds of vegetables and plants and seaweed and rice, fermented rice. Is that a reason that comes into account why this gene development takes place?
So, okay, yeah, the two separate things, right? The things like, as you say, that's in your genetic code or how the genetic code is read about, and the other portion is like the environment or what you do with it, right? So one thing that has been found through the animal kingdom is that if you starve animals, so they're not malnutritioned, that increases their lifespan. And so these studies have been really like started in like 1935, where they looked at rats. And the question there was like, if you feed the rats lower amounts of food, like 40% lower, you would assume that they have all like detrimental effects and like that. But the contrary was seen, right? They're all fitter and mobile for longer and they lived longer, right? And then people have tested like in any animal you can think of, even spiders and things like that.
There must be a critical value, like, of course, at some point they will start being malnutrition.
Yeah, exactly. Okay. So there's a critical value. So if you just have it right, you can benefit from that, right? So it's a nutritional control, and what you eat, right, is also perceived. So again, you eat something, your glucose spikes, right? And your glucose levels are regulated by insulin and HF1 levels, right? Which the mutations are found, right? So it's all these nutrition sensing pathways that are involved, that if you tweak them genetically, it makes the animals live longer. Or you can tweak them by, either reducing food intake or change what you're doing. Now, coming back to the Okinawa, which is a really good example, because if you take those people and put them in a different environment, so some of them migrated to the US, right, where the lifestyle is changed, it completely abolishes like their benefits they're having, so they're not long-lived anymore and things like that. So
does it mean it's really the environment they're in that affects their lifespan?
So the lifespan they have, right? They have, as you mentioned, they have the diet where they eat. They never overeat, so there was little portions, and it's also very healthy what they eat. But they're also very active, right? So also they're like older people, they're socially really active. They start learning new things, like, you're 80 years old and they start learning how to ride a bike or things like that, right? So they start trying to new things, and every day they have a routine where they do physical exercise. So they actually do what your doctor tells you to do, right?
Yes.
And that's really like, it seems to be beneficial.
It does sound like if you follow everything your doctor or self-help books tell you to do, you might just end up living a lot longer than you usually do.
Yeah, I mean, at the moment, so far, it's that what we can do, or it's the best thing we can do at the moment.
It's what
right?
we have control over.
Yeah, and so I'm not sure whether it's the goal to live longer, but to live longer healthier, right? That's the real goal.
Right. And so does that mean that from this extension of life is not necessarily genetic, it's more just lifestyle? Or does it affect the genes?
It also affects the genes. So you change, of course, again, your gene expression. When you start, like, you change your lifestyle, it also changes gene expression. So it goes hand in hand. So, I mean, scientists believe now that there are like 3 major things that influence the lifespan, right? One is the genetic setup you have. And then the second thing is the environment. So you change the environment. And the third one is randomness. So there's always some randomness into it, right? So these are the 3 things. And so, but of course, the environment affects your gene expression, and the gene expression, can affect the rest.
Okay, now let's get to the science. Could you tell us simply, but in detail, what is it exactly that you do? Because you talked about genes and DNA and neurons and cells. What is it that you look at in the lab?
Okay.
That's very confusing.
So in the lab, we have 2 approaches, right? We have an unbiased approach, and we have a hypothesis-driven approach. Okay, and the model we're using is this little roundworm C. elegans, because it's it lives for 3 weeks, and the genetics very easy to manipulate. So in the unbiased approach, we ask the animal actually what gene mutation increases their health and their lifespan. And we can do this in a high throughput fashion. So these animals are 1 millimeter in size. So you can have on a small plate, 6 centimeter, you can have thousands of them, okay? And then you can induce mutations with some chemicals. Random mutations, they can hit anywhere in the genome. And then we're just trying to figure out like which of those mutations, right, actually increase the life and the health of the animal. And then we're trying to go back and find out what is the mutation, right, in which gene. And when we know the gene, we're trying to figure out the pathway. Once we have the pathway, we're trying to figure out the molecular mechanism. And then we're trying to see whether that holds up in a higher organism, like in mice. And then in the end goal would be like something beneficial for humans.
So if I try to create an analogy out of it, tell me if it's correct. You're just randomly throwing balls at a wall, and you see at which point when the ball hits, the ball retraces the farthest, or like the wall does something. And then you look at, okay, so when you hit the ball here, this happens, and then you try to reverse engineer that thing, and you focus on that bit.
Yeah, exactly.
Okay.
Yeah.
Thank you for listening. If you liked it, we'd really appreciate it if you shared it and gave us your feedback. This was part 1. Part 2 comes out this Thursday. For more details, visit our website simplified.xyz.